Hello friends, welcome to Hottest Botanica, that is your botany classroom. Today we will discuss a very important information of chapter biological classification. Topic is virus. Yes, this is very small but interesting topic of unit diversity of living world. You all know in our previous video we discussed this Q-Sep, that is question, solution, exploration up to the perfection. It will help you to understand the basic meaning of the question, the sense of the question, so that you can solve any question from the same theme. So today I have taken this question from AIPMT of 2015. So in this question you have to find out wrong statement. So yes, we have given the 4 statement. Out of that, 1 is wrong. Yes, I know it's a very easy question. You can easily find out. But I will go for Q-Sep as I discussed. We will discuss all the concern of this question, their theme, and all concerns. So, according to this question, the wrong statement among these 4, we will go each and every statement in detail. So the answer of this question is the second statement, and that second statement is the wrong statement, and it was viroid was discovered by Ivan Voski. Yes, it is wrong statement because the viroid was discovered by Theo Denner, not Ivan Voski. Yes, Theo Denner discovered this new infection agent which is smaller than virus, and it is viroid. And first disease caused by viroid was potato spindle tuber disease. Yes, you all know. Now come to explore this question. Yes, exploration and perfection of this question. Source of this question is your NCERT. Yes, it is taken from page number 26 to 27. Yes, this is moderate type question. Chapter involved is biological classification. Topic is your virus. And these are the concern, these are the statements from where this question has been derived. So I will detail this all statement in a very good flow chart, so you can easily learn and answer any question regarding this subject. Now see the first part I discuss. First, you must know about the concept of component of virus. Yes, whenever you talk about component of virus, so first of all, the complete virus is called viroin. Means to say, it has nucleic acid. It is being covered by its proteinous coat, that is capsid, and it may be of envelope. So envelope may present or absent. The nucleic acid may be either RNA or DNA. Any one should be there. Both cannot be present. It may be the part of your question. And the example is SP hema. Yes, SP hema is a very good example of your animal virus. And MLCV, dwarf and stunted, this is very good example of your plant viruses. I will go in detail in next slide. So the first of all, this Theo Denner, yes, see the great scientist. I think you have not seen till now. You can recognize our great scientist. Yes, Theodor Otto Denner, and he is the great scientist who first discovered this viroid. So in viroid, what we have? Yes, we have only this genetic material. This genetic material is usually DNA. So there is no protein, there is no envelope, only there is nucleic acid as RNA, and this RNA is infectious. It will cause disease. It is free RNA, single stranded RNA having low molecular weight. And first of all, the discovery of viroid and their disease was first disease viroid is in potato, that is potato spindle tuber virus. So what do you think? If the viral component have only protein, there is neither envelope and nor nucleic acid. So if there is only protein, there is no nucleic acid and envelope, then only protein, and this intact protein is called prions. Yes, and the prion is highly folded. So remember, it has only protein, but its size is not smaller than virus, rather very similar to virus due to its folded structure. So very good example we have is BSE. Yes, this is also called cow mad disease. This is bovine spongiformis encephalopathy, and this was first caused in cattle. And from that cattle it come to the human and showing the same symptom. There is the neurogenetic diseases where brain get very injurious there in the our brain, and there is the holes, and due to the holes the thinking power, balancing of the body is being disturbed, and finally it becomes lethal. So it was first of all noticed in the cattle, later on it was noticed in human, and it was called as Creutzfeldt-Jakob disease. So that's why both is the analogous variant of each other. Now, so I think from this slide you are very well understanding that how we have three important parts: complete viral particle. Yes, the complete viral particle, we have nucleic acid, we have protein, and the envelope present or not present. As in TMB, no envelope is there. In AIDS, envelope is present. Now, in viroid, what happened? Yes, we have only genetic material. There is no protein and no envelope, and only protein is there, and this protein is highly folded. That's why its structure is very similar to the virus, and this is nothing rather than prion. Right, friends? Now, yes, what was our third and fourth statement? Third statement was W.M. Stanley showed that virus could be sterilized. This was very important discovery. That's why he got Nobel Prize for this very noble work. And before that, yes, this statement is also correct, that M.W. Wizardnicki, yes, M.W. Wizardnicki, he first time coined the term contagium vivum fluidum, means infectious liquid in disease, that is causing diseases. Now try to go in detail. See, yes, we have three important scientists here. I think you have not seen before. Recognize now. It's very easy to recognize the name of the scientist by their figure. Yes, so we will talk about the discovery of virus. First of all, I will talk about the recognize. Who first time recognized about the existence of virus? Yes, he is our DJ Ivanovsky. Yes, Dimitris Zosvikovics Ivanovsky. So he found these microbes which is smaller than virus. That time if you are stating the statement smaller than bacteria, it's very tough to say, because at that time what happened, a jar were given that if you have any infection, then you must have this filter paper. So this filter paper, what? This is the bacteria proof filter paper that will extract all the infection. So if you have any liquid, so if you will, this liquid will pass through that, what happened? Bacteria will filter. So what happened? Once Ivanovsky, what he found, he extracted and passes through this bacteria proof filter, and he found the liquid is coming. And this liquid is, of course, causing the disease in tobacco, which is Mosaic disease. And since it has passes through this filter, so of course its size is smaller than this bacteria. So he called, we have a very good microbes whose size is smaller than bacteria and causing the disease. This was a very great discovery. So it need a very great courage and good repeated experiment to state this very big thing that we have some microbe which is smaller than bacteria. But our Vizarnik took this step, and he understood the work of Ivanovsky. Yes, and what he did, he extracted that very infection of mosaic from tobacco, and he found same result, and he caused, yes, infection is being again caused in the healthy plant. Means to say, the healthy plant is being infected by that very liquid, and that liquid has some infection which is smaller than bacteria, what the Ivanovsky was telling. So Vizarnik told, yes, you are right, and he gave a statement, and he told what? Contagium vibum fluidum. Contagium infectus, vibum living, fluidum liquid. So we have liquid which is living, causing the diseases, infection on the plant, healthy plant. So we have really a microbes smaller than bacteria causing that disease. But what should be its structure? What should be their structural component? It was not being explained by Vizarnik. Later on, the great scientist W.M. Stanley, yes, Wendell Meredith Stanley, he crystallized that very liquid, and he found that liquid contains large of protein, not only protein, rather nucleic acid and protein. So he found that there is the nucleic acid and protein, so nucleoproteinous particle is there in a very crystal structure, and that very structure whenever come on the host, it will cause disease. So at that time he found that crystal structure, he showed that very crystal structure to the world, and recognized how there is a microbe which is smaller than bacteria. For that very noble work, he got Nobel Prize and honored as father of virology. So friends, by this way you have to remember the first scientist who recognized the virus, yes, DJ. So you will start this sound, dance, music, DJ. Yes, DJ is Ivanovsky, DJ master is Ivanovsky, who sound the recognition of virus, and the first song was sung by our this Vizarnik, yes, Martinus Willem Vizarnik, and he gave the song that is Vibum Contagium Vibum Fluidum, and the whole credit was taken by Stanley, yes, who sought scientifically the crystal form of virus, and in this way the virus was discovered. So I think it's very much clear. You have to remember who discovered, first time who recognized, yes, Ivanovsky, DJ master Ivanovsky. What he did, he just took the bacterial filter paper and found that there is a microbe which is smaller than virus, so it's very infectious. It was repeated by Vizarnik, crystallized by Stanley. Vizarnik, MW, just opposite, MW, WM, MW, WM. So these are very good dancing. You can say Stanley dances, getting this Nobel Prize, and Vizarnik give the sound. What was the sound? What was the song? Yes, song was, yes, Contagium Vibum Fluidum, and the DJ, the first sound, first precursor of the discovery was Ivanovsky. So friends, students, you can very easily understand now who recognized, who demonstrated, and who showed first time about the virus. By their diagram, you can also very easily recall these three eminent scientists. Now, the first statement was Mosaic disease. Yes, this mosaic disease in tobacco and AIDS in human being are caused by virus. Yes, this statement is also correct. But I would like to give you a very good mnemonics to remember all the examples given in our NCERT, because that may be the part of your coming examination of Neet. Yes, whenever you need to remember the example of animal viruses, you should remember SP Hema. Yes, Hema, you call Hema, and this Hema is SP, so you can remember SP Hema. So smallpox, yes, SP. H for herpes, I for influenza, M for mumps, and A for AIDS. So you have to recall this mnemonics SP HEMA for animal viruses. So we have here 5 important examples. Similarly, if you want to recall the examples of virus in plants, so you have to recall very good mnemonics MLCB, dwarf stunted. MLCB, dwarf stunted. M for yes, mosaic formation. L for leaf rolling. C for curling. B for vein clearing and alloying, and stunted virus. So we have the 5 important examples. SP HEMA in animal viruses, and in plant viruses we have MLCB, dwarf and stunted. So in this way, students, you can very easily remember the 10 important examples of viruses, 5 from animal and 5 from plants. So this is the very good Q-Sep of your this very important example. So what we studied today, yes, example of the viruses. In animal viruses we have SP HEMA. For plant viruses we have MLCB, dwarf and stunted. Yes, we remember 3 important scientists recognized. Ivanovsky, DJ Master Ivanovsky, recognized by, yes, very good song, his song. What is the song? Yes, Contagium Bibum Fluidum, given by M.W. Wisanek, and the well credit gone to the great scientist Wendell Meredith Stanley, who got a Nobel Prize and honored as the father of virology. And see, the Theodore Otto Denner, who discovered viroid having only nucleic acid as RNA, which is infectious free, single stranded having no molecular weight. And this is the source of the question. This is your answer, solution, question, and this is the whole about this very important question and explanation. So I think you like this the whole statement, and if you want to such type of the explanation of the question, then you can write your comment, your suggestions and good comments and your requirement. Thank you very much, friends. Thank you very much.